So Anne is also a director of research at CNRS. She spent most of her career in Montpellier, but also spent some time working with Ben Sheldon at Oxford, Tim Clinton-Brock in Cambridge, and then with Denis Réal in Canada. She has a huge track record working on mésanges, blue tits and great tits, and I think she's going to talk about how they live in our cities.
Yes, thank you very much for the introduction. Thank you to Virginie and Abderrahmane for inviting me to give this talk today. Okay, so I am indeed going to talk about adaptation of birds in a city environment. So in my group, we've been studying for the last 2 decades at least, rapid evolutionary and plastic responses in a world that is changing very rapidly. And in particular, we've been exploring how bird and different bird populations of different species adapt to climate change, how warming and extreme climatic events can trigger new selection pressures, and how this can trigger evolutionary and plastic responses. These studies we've been exploring mainly in pristine environments, such as these forests in Corsica. But I want to translate you today from these pristine environments to a very different theater of studies. And first I want to mention that so we do this work by modeling the different underlying processes, such as gene flow, drift, and genetic variation and epigenetic mutations. And we also integrate the complexity of the environment. And so again, the environment that we've been studying are mainly forests and natural habitats. But today I want to translate to a very different habitat, which is the city environment. Here, a picture of my hometown of Montpellier. And so what we've been discovering kind of in the last decade or so is that cities are a really interesting environment to study these evolutionary and plastic responses. And this is because they have a high concentration of factors that could influence these different processes, and in particular selection. So cities are in particular highly fragmented. I am not sure at all where I didn't test that before, sorry about that. So cities are highly fragmented. They show high heterogeneity in time and space, and they have what I will call here a lot of environmental stressors for most of wildlife. In particular, there's the urban heat island effect, whereby temperatures are much higher in cities. There's air pollution, water pollution, disturbance by humans and their pets and cars, light pollution, and sound pollution. And so this means that we are expecting that cities will be an environment where evolution should happen quite frequently. And I want to give one example only in this introduction. And this is a story that probably a lot of you have known and read about. The study was initiated in the London Underground, even though it could be in any underground system of the world. The Underground in London, as many tube systems, is an environment that is highly fragmented because of these different tunnels that are not necessarily connected. There's a strong heterogeneity, especially between what is happening underground and what is happening above ground. And there's a very strong heat island effect, whereby temperatures are really, really high. And this has led to the evolution of what is said to be a new species of mosquitoes, Culex molestis. So Culex molestis has evolved from Culex pipiens. Because of high fragmentation of this tube environment, they have evolved toward a very different sexual reproduction, whereby males and females create pairs instead of swarms of insects. Because of the human presence and also possibly the rat presence, this Culex molestis has evolved towards a preference for mammalian and especially human blood, and this is why it's called molestis, because it molestes us all year round. So while Culex pipiens is more attracted by bird blood, and because of the urban heat island effect, this species has dropped the diapause. So there's a strong change in the lifestyle of this insect. So this is just one study that I wanted to highlight to show how promising cities are in terms of very altered environment where we can see rapid selection. These kind of studies we've reviewed in a book that came out in the worst possible time to publish a book. That was April 2020, when it was said that all conferences would be canceled for the next 2 years. So in this book, we especially have a chapter entitled Adaptation Genomics in Urban Environments, which I wrote with my postdoc, Charles Perrier. And this is pretty much a roadmap of how we can go about investigating urban adaptation. But this can be applied to pretty much any environment where you find strong phenotypic divergences. And the previous talk by Vincent Collot made a nice introduction of this leitmotif example that I'm using here of the Carbonaria morph of the pepper moth. So I'm using this here just as an example of where we usually start off when we're interested in adaptation is witnessing differences, for example, in morphs. And as an evolutionary biologist, the first question I'm going to ask myself, or one of the first, is whether these different morphs are adaptive. The second question will be about whether they are of genetic or plastic origin. And then if you have the time and the money and the resources, you can dive into these genomic section of the work where you can investigate which genes are involved and where they're located and how much genetic influences there are. So this roadmap is what I'm going to use as an outline for my results on the study of great tits in cities. And so we're starting with investigating phenotypic variation. So first of all, I want to acknowledge this is very much a collaborative project. It was the case from the start, and I want to acknowledge the work especially of Arnaud Grégoire and Marcel Lambrecht, who were on this project from the start. And most of the results I'm going to present are from the postdoc of Charles Perrier and PhD of Odd Kezerg and Megan Thompson. Also, I will briefly tell you about a little bit of a crazy experiment we did in spring 2023, where we decided to hand raise chicks or eggs that were coming from the urban and the forest environments. And so this involved a lot of new people in the project, and especially the Montpellier Zoo nursery that helped us raise these chicks from 7 a.m. to 9 p.m. every day for 2 months. Okay, so the Great Tit project in Montpellier actually started with the Blue Tit project, which was initiated in the 1970s by Jacques Blondel. And we have a forest in the north of Montpellier called La Rouvière, and this is a snapshot of this forest. Each dot is a nest box, and we've been studying blue tits and great tits there since 1991. In the city, we erected nest boxes in 8 different sites. You have one site here, and I'm putting this as an illustration to show that we have nest boxes in parks, but we also have nest boxes, and you don't see the dots and numbers here, but along roads and in very industrialized areas. So in the city of Montpellier, we set up these nest boxes a little bit more than 10 years ago, and we've been monitoring the nest boxes in the forest and in the city. It's very important for us that we have a whole gradient of urbanization and not just one forest and one city area. So here you see the 8 different study areas in Montpellier. So within these areas, there's a gradient with parks and roads, but also across the gradients, across the sites, there's an urban gradient with some sites like the zoo that are quite natural and other sites that are much more urbanized. So in terms of fieldwork, what we do is that we capture all the birds that breed in these nest boxes, and we take some classic morphological measures, such as the size of the birds and the size of their wing and their tail. We have classic life history traits as well. When do the birds breed? How many eggs they lay? How many hatchlings and fledglings? And then perhaps a little bit more original to our monitoring is the measure of several behavioral traits. And in particular, I'll give you results about the exploration score. So a brief explanation here. Right after the bird is captured, it is placed in a little box inside here where it can rest. And then after a few minutes, it is released in this part of what we call the open field cage, where the bird is free to explore the cage. And here is a camera that is filming the behavior of the bird. You will see a little video later on of what's happening inside this open field cage. Okay, so what do we find in terms of phenotypic divergence between city and forest birds and along the urban gradient? I'm showing here for simplicity the comparison between forest and city. And so what we find is that basically every trait that we measured on these birds were different between forest and city birds. So city birds are smaller, they are lighter, and they have shorter wing and tail lengths than their forest counterparts. In terms of life history, they lay around 4 days earlier in the city, and that's in Montpellier. In Paris, it's even more drastically different. They have around 2 eggs less in their clutches, and they have a smaller number of fledglings and hatchlings. And in terms of behavior, they are more aggressive, They explore this open field cage much faster, and they have a faster breath rate when they're under constraint in our hand. So again, I just want to mention here that I'm just showing a comparison between forest and city, but if I did similar graphs along the urban gradient, then we find similar results within the city. And previous work and ongoing work from our group and other groups have shown that all these traits are heritable. It is even more so the case for morphology, but even the behavioral traits are heritable. So around 30 to 40% of the variance in these traits are due to additive genetic effects. So of course, our next question is here, we see a strong divergence in traits that we know are heritable. So does that mean that there has been adaptive evolution between the forest and the city environment? So before diving into this question of adaptation, I want to scale up a little bit these results on the fact that birds are taller in the city and show you that because the grey tit is also a model species, not as much as Arabidopsis, but it is a true model species for ecology, we have a lot of replicates. So PhD student Megan Thompson used here 13 different areas where grey tits are studied in the forest and in the city, and she conducted a meta-analysis. And as you can see here, the log response ratio is for traits that are morphological traits. So each dot is one morphological trait compared between urban and forest birds. And there's overwhelming evidence that the urban birds have a smaller morphology. But another result that we found with this data, and this is something I'm going to swipe through, but if you're interested, come and discuss this, we find that urban birds have larger variances. So there's larger phenotypic variances in the city, and this is something that we want to explore in the future. So Megan wrote this review paper on how variances can differ in urban environments, and it's something we really want to explore more in the future. Okay, so we see very strong phenotypic divergences. Is it adaptive? This was the work of Aude Kayser during her PhD. So here I'm just reminding you that birds are smaller in the city, and we're now going to look at what selection is saying about whether this is adaptive. These are selection gradients. I'm going to walk you through it, but basically when we find a significant selection gradient like here, the little blue star, it means that, so here we have forest males, the circles, sorry. So forest males have a significant positive selection gradient for their tarsus length. It means that being taller in the forest is selected via the higher reproduction of these males. So in terms of morphology, this is the picture that we get. Pretty much no or very few selection gradients that are significant, and they're only significant in the forest, selecting for lighter males and taller males. So we don't see that being smaller in the city is favored by selection. In terms of life history, a reminder here, birds lay earlier, smaller broods. And what do we see in terms of selection? Well, even though birds in the city lay earlier, they are not selected to lay earlier. They are selected to lay early in the forest only. And even though birds in the city lay smaller broods, they're actually selected to lay larger broods. So here again, pretty much like the morphology, what we see is that we don't see selection favoring the traits that we see in the urban environment. And I'm going to spare you the details about the behavior, but what we find is that basically all the behaviors that are displayed by birds in the city, such as high aggressiveness, high speed when they explore a novel environment, this is all associated with lower survival. So overall, what we see is that the observed phenotypic divergence that I've illustrated before is not due to an adaptive response. So it might be maladaptive. It might also be that it was adaptive in previous times, and we can't measure past selection right now. Okay, so the next step is to test whether the differences that we see are of genetic or plastic origin. And for this, one of the best approach is conducting experiments such as common garden approaches, where you collect individuals from the two environments and you raise them together. Now, this is really difficult to do with vertebrates, but we still try to do that. So what we did last spring is that we collected eggs in the city and in the forest along the urban gradient. These eggs were incubated by foster parents, because it's really difficult to incubate eggs in the lab, and especially to raise the chicks in the very first days after hatching. And right after hatching, the eggs, the nestlings, once they were thermoregulated, they were placed in a nursery where we hand-fed them and raised them, and they are still in aviaries right now. And so what we're doing now, and we've been doing for a year, is phenotyping these birds for all the traits that I mentioned previously. So the morphology, the physiology, behavior, and cognition. I'm not going to show you details on this because I wanted to spend more time on showing how we integrate genomics and epigenomics approaches, and also because the results are still very much ongoing, but I will show you results on 2 traits. So the first one is respiratory rate. So these are the results here for forest and city birds caught in the wild. And as I said before, we have, so this is number of seconds it takes for 30 breaths. And so we have here urban birds that are faster, that have faster breath rates. So what do we see on these birds that are hen-raised? Well, we see a very similar difference, albeit a shift. So what you can see is that when the birds are hen-raised, they have slower rates when you handle them, probably because they're used to humans and so they're a bit less stressed. But there seems to be still a shift in the sense that birds that are originating from urban eggs still have a faster breath rate than birds originating from forest eggs. Now, a very different trait, and now you can see this video of the open field explorations. So the bird has entered from here, and we're video recording it for 5 minutes. And so some birds stay very still, and others will hop all the time. And so our field work in the wild has shown that urban birds are faster explorers. What do we see with these birds that are hen-raised? Well, there's no more any trend. So this suggests that the shift that we see between behaviors in the wild between forest and city might be due to plasticity and might not have a genetic background. Okay, so now let's dive into the genomics. So the genome of the great tits is somewhat bigger than genomes that we've explored this morning, especially Arabidopsis. Also, I didn't have funding to do the whole genome sequencing. We had some candidate genes that were involved in the traits that we were interested in, but we were interested in such a wide variety of traits that we actually decided to go via a different route, and this is especially rat sequencing to look at the whole genome, but different small parts of the genome. This is a collaboration with Marta Solkin in Warsaw and Juan Carlos Senar in Barcelona, because we sampled birds from these 3 cities and 3 forests that are nearby the cities. So we sampled great tits in Warsaw, Montpellier, and Barcelona, and we did rat sequencing and RRBS sequencing for methylation patterns. Okay, so the results on this. So here I'm showing the redundancy analysis where every dot here is one individual. I've kept the same kind of code that circles are males and triangles are females, and you have 1 color per city. So the first thing, obviously, that is very obvious here is that individuals, they cluster by city. So yeah, for those of you that are not used to RDA, they're very similar to PCAs. So the closer 2 individuals are, they're more similar, their genomic background is based on this rat sequencing data. And so you can see that there's an obvious clustering of birds within cities, which was clearly expected, obviously. So the first and second axis of variation are the latitude and longitude. But we do have a third axis of variation that explains 2% or 2.2% of the variance. So it is small, but it's significant in the sense that we do see some genomic differentiation between forest and city birds. So you can see that because the open, so for example here in Warsaw, you can see that all the white dots are kind of clustered in one area and all the orange ones are in another area. So these are forest and city birds. So there is some genomic traces of selection of the urban environment. In terms of methylation patterns, the picture is quite different because actually the first axis of variation is sex, and this is a classic result that males and females differ substantially in their methylation patterns. So this is what we find again here. So you can see that the axis 1 is having all the males on the one side and all the females on the other side. There's also a clustering by city that you can see. So I didn't represent RDR, the axis 2, but it was a clear differentiation between cities again. And then again, the third axis of variation is this urban-forest contrast. But we don't have much more variance explained that we can see at the genomics level. We have, again, 1.9% of the variation in the methylation patterns that is explained by the urban versus forest origins of the bird. And to give you a little bit more details about this methylation patterns, here I'm representing what we call the circus plot. So you have the whole genome of the grated with, first of all, the macrochromosomes, so chromosome 1, 2, 3, etc., and then it goes towards much smaller chromosomes. And here you have represented the differentially methylated regions for Barcelona, Montpellier, and Warsaw. So the red dots are areas, so DMRs that are hypermethylated in urban birds, and the blue dots are areas that are hypomethylated in urban birds. And there are two main conclusions that I want to bring you to here. The first one is that if you take one here, for example, hypomethylated area in the genome of birds from Warsaw, you will most probably not find this DMR in the two other cities. So of course we have only 3 cities so far, but it's an indication that there is no strong evidence for parallelism in these methylation patterns. And the second thing is that we kind of expected hypermethylation to be associated with the city environment because we know that pollution can lead to methylation, and this is not what we find. There are not more red than blue dots in this circus plot. So what we're doing next is exploring where these DMRs are located and whether they can be associated with interesting genomic regions that we know are related to candidate genes, especially for the behaviors. Okay, so I'm reaching my conclusions here. So overall, what we've seen is that when we study birds across an urban gradient, we see very strong phenotypic differentiation between the forest and the city, but also along this urban gradient. It does not seem to be explained by selection as we can measure it now. So all the traits that we've seen that are characteristic of urban birds are not associated with higher survival nor higher reproduction. The effect of urbanization can be seen at both the genomic and the epigenomic level. It's largely polygenic, and we could have expected that because we see that many, many traits are involved, and all these traits are quantitative traits. And for what we can tell from now, with 3 cities that have been explored, there doesn't seem to be any strong parallel response to living in the city. And then finally, the common garden experiments, which we are still analyzing, shows that some of the differences that we have witnessed in the wild are maintained in these common garden conditions, so might be due to evolution, but not all of them. And I want to finish with this slide. For those of you that are interested in studying eco-evolutionary process in urban environment, this is really a blooming field. And last year we conducted what we call a horizon scan. So basically we asked everyone interested in urban eco-evo to give us the questions that they feel are the most exciting for the future. We had 700 questions that we gathered in 30 big questions that we classified in these 6 different areas. So this is basically the future of urban eco-evo. In our group, we are particularly interested in exploring more the underlying processes that are involved in adaptation to cities. And I have a new project where we're trying to combine the knowledge that we have on adaptation to climate change with adaptation to urban environments. So again, we had only 1 coffee break so far, but a lot of fruitful discussion in 1 coffee break. So I invite you to come and chat with me if you're interested in these questions. Thank you very much.